All right, guys, so this is going to be chapter 13, cardiac functions. So we're going to look at the heart and how the heart works. So following along with your outline, if you need to review the heart on your own, so review the anatomy of the heart and the major vessels and structures of the heart, and that would be 13.1 through 13.3 of your textbook. But we're going to start with the conduction system of the heart. So this is just a review image of things that you want to just sort of keep in mind, review the structures of the heart. Okay, so the conduction system of the heart is created by these cells called autoregulatory cells. And the autoregulatory or autorhythmic cells, right, they create their own rhythm, is going to be nodal tissue. So you guys know this as the SA node and the AV node. And it's also going to be something called conduction fibers that conduct the action potential throughout the heart. And that's going to be your bundle of His and your Purkinje fibers. So it turns out that, so autoregulatory or autorhythmic cells can create their own action potential, which means that their membrane can depolarize spontaneously. And these are actually located all throughout the heart, but they're concentrated in areas of the SA node and AV node. So they're called pacemaker cells. And the reason why the SA node is going to be the pacemaker, right? If you remember from anatomy, the SA node is the pacemaker of the heart, is because it creates the fastest action potential. So the firing rate is the fastest. Therefore, it kind of wins the race. So it depolarizes first, and it's going to set the entire heart's rhythm. So the SA node is going to set the sinus rhythm. So the regular heartbeat is called sinus rhythm, set by the SA node. If the SA node fails, then other nodes can pick up, and so can the conduction fibers. So the AV node can pick up. It has a slower than normal heartbeat. So if you do have an SA node problem, the AV node takes over and you have a slower than normal heartbeat. And if the AV node also fails, then the bundle of His and Purkinje fibers can also actually produce a heartbeat, but it's going to be much slower and actually pretty dangerous. But these other, conduction, these autoregulatory cells can actually create a heartbeat. So moving on to the number 4 in the first part of our lecture, keep in mind that the SA node actually has a really quick firing rate. And this is actually the at rest, your heart rate. This is pretty fast for creating that heartbeat. So the SA node has the potential to go up to 100 beats per minute, which is very, very fast. So externally, the vagus nerves, the autonomic nervous system does come in and slow down the SA node. And also it does respond to hormones. We'll look at that later. All right, so just looking at number 5, before that picture, there's a pause before we get to the AV node. So the SA node depolarizes, and then there's a pause before the AV node depolarizes, and this is because we want the ventricles to be able to contract. We'll look at this in a little bit. And the reason why the AV node, the conduction moves through the AV node a little bit slower is because the cells are actually tiny, really, really tiny cells. So they actually don't conduct impulses as quickly. And so the message from the SA node, when it gets to the AV node, as it creates its action potential, it's slowed down. And so the rest of the fibers, the Purkinje fibers and the bundle of His, opposite, right? The bundle of His would go first, then Purkinje fibers, they get the message in a delayed fashion. And that's good because it allows the ventricles to fill with blood before that they contract. All right, so let's take a look at our picture of cardiac muscle cells before we move on. So cardiac muscle cells are linked by gap junctions, and this is going to allow the conduction to go through the muscle fibers really, really quickly. So we can see that in the regions of our intercalated discs, right? These are the dark staining lines that you see in cardiac muscle tissue. This region has both gap junctions. So we have basically connections of the cytoplasm. So the electrical signals spread very, very quickly from one cell to another. But we also have a second structure at these areas called desmosomes. And desmosomes are junctions between cells. So they're proteins that hold the two cells together. And desmosomes are specialized to have these fibers that reach into the cell to provide more structure, because these cells are going to be contracting every second for your entire life. And so there's a lot of stress happening between the cardiac muscle cells. And so these desmosomes hold them together, and it can resist that mechanical stress. All right, so looking at the conduction system of the heart. So this should be quite a review. So the sinoatrial node, this is going to be our pacemaker, which is going to fire first, create its own action potential. That action potential will flow through internodal pathways. So these are going to be conduction fibers that run to both atria. So the two atria get the messages first to fire or to contract. Then the message goes to the AV node, and this is where it slows down, right? So the atrioventricular node has tiny cells. It slows down the conduction, but when it fires an action potential, it'll flow through that bundle of His, right? And there's two bundles. Remember, there's a right and left bundle branches. And those go down to the apex of the heart, at which time you're going to spread into smaller Purkinje fibers. And those Purkinje fibers will come up. So the message goes down to the apex first, and then it will move up towards the base of the heart from the apex. All right, so that's just a quick review. So again, realize that the atria contract first, right? Because this depolarization happens first, so both atria contract. Then there's going to be the ventricles contracting, so both ventricles would contract second. All right, so let's move on to the pacemaker potentials. This is just a wrap up, sorry, I didn't realize I had this. So interatrial pathway, right, we have simultaneous contraction of the left and right atria. This is very quick. And then we have the internodal pathway from the SA node to the AV node. The AV node is going to slow the conduction, and it's going to cause the ventricles to contract after the atria, right? So we have that atria contract before the ventricles. All right, so this is just another look at the action potentials as they spread through the heart. So we have our action potential in yellow. So first our SA node fires. Action potentials are conducted from the SA node to the atrial muscle. As they spread through the atrial, right, the action potential spread through the atria to the AV node where the conduction slows. So there's our AV node. Then it goes down the bundle of His, both left and right. Action potential spread from the apex towards the base of the heart, and eventually the heart is in this relaxation stage. So eventually the entire heart returns to resting, remaining there until another action potential is generated by our SA node. All right, I think we need to look at our pacemaker potentials first. All right, so we're going to be looking at two different potentials. One is for the pacemaker, so the pacemaker cells, right, the SA node and the AV node, how they depolarize. And then we're going to look at the contractile cells. So these are just going to be the myocytes, the muscle cells that contract in response to the action potentials coming from those nodal tissues. So first, let's look at what's happening in our pacemaker potentials. So you might be asking yourself, how is it that these pacemaker cells can set their own heartbeat, that they can depolarize on their own? So that's what we're going to answer here. So pacemaker cells don't need an external stimulus, right? They can depolarize on their own. And this is because they have an unstable resting potential. You can see that the -60 is going to be our sort of baseline resting potential, but they don't stay there for very long at all. So let's just follow the outline, right? As soon as we have depolarization, there is a slow ramp back up to threshold. So let's just start right here, right? We're starting at resting. So notice that we don't rest for very long, and there is a slow increase in membrane potential. So we're going from negative to positive, right? So there's a slow creep of positive ions coming into the cell. This is going to be our sodium ions, and there's a slow influx of the sodium ions. This is due to a channel called a funny channel, which is a funny name, I guess. So let's go ahead and put, this is due to our funny channels, because scientists, I guess they wanted to be funny, and they named it funny because they were behaving in a funny fashion. So they kind of just open after repolarization happens, and they allow sodium to come into the cell. And what happens is they build and build and build very slowly. I mean, slowly is relative because you can see the time frame is just about 1 second. Until they reach this potential that is right below threshold. And then right below threshold, there's going to be another influx. So calcium channels are going to open, right? So we have our slow influx of sodium. That's going to cause, at one point, right before we reach threshold, it's going to cause our calcium channels to open. Calcium is going to come into the cell, right? Move into the cell to make that cell even more positive. And so this happens very quickly. So you can see this rapid red area where we have an increase in our membrane potential, positive. So that is going to be our action potential. And then we're going to sort of go back, right? We got to repolarize the cell. And this is going to be familiar because when we repolarize the cell, we're going to have an outflux of potassium channels. So potassium channels will open. And then at this point here, right? So potassium channels will open, and then out goes potassium, and there's a whole flood of potassium leaving the cell very quickly. And so we go down and we go back to resting. Okay, so this is, we're going to repolarize and because of our potassium channels. Okay, and everything starts over again. So the reason why pacemaker potentials, they can create their own action potentials is because they have an unstable resting due to the fact that they have these funny channels that allow a slow leak of sodium into those cardiac muscle cells. So that slow influx of sodium creeps up until we open up those calcium channels. And then calcium channels is what really drives the depolarization. Okay, so that's how pacemaker potentials work. Now let's look at how your contractile muscles work. So just the muscles of the heart that contract. So these are going to respond to the voltage that's coming from externally, right? They're going to respond to the action potential set by your pacemakers. So we have some differences here. So if you notice, there is a different kind of picture, a different shape. And this is really due to this large plateau. Okay, so we'll talk about that in just a second. So there is a rapid depolarization followed by a plateau, and then we have repolarization. And you can see how this area is very gradual, right? The plateau extends your action potential, and then your repolarization is very gradual. What this does is it really gives you a long refractory period. And this is super important for your heart to have this long refractory period, because what I have in the notes, I'll just paraphrase, but you want the heart to contract fully to eject all of the blood out into either the aorta or the pulmonary trunk. If you have the heart partially contracted and the blood is moving out, and then all of a sudden you set another heartbeat, so there's another action potential set, you have two contractions very close together, and that's not going to be an efficient way to eject the blood out of the heart. So you need this extended refractory period, because if you have these premature contractions, then you're not going to have enough blood leaving the heart, and that's not going to be compatible with life, which is what's in the notes, but you're not going to get enough oxygen or blood to your organs in this way. So this long refractory period really helps with keeping that blood, the ejection of blood, efficient. All right, so when the action potential, so we have the voltage coming, right? And we have our action potential. So this is pretty familiar. So we have our voltage-gated ion channels, and we have a massive influx of sodium causing our positive membrane potential. And then what happens is the plateau is due to the fact that we keep a positive membrane potential, not because of sodium, but because we're opening other calcium channels. So now we have these slowly opening calcium channels with this depolarization, and the influx of calcium is going to keep that positive charge high. All right, and so these slow calcium channels will start to close eventually, and then you're going to begin repolarization, and repolarization is the same as what you're used to. We have the potassium channels opening, right? So potassium channels open, potassium will leave the cell, driving the membrane potential back towards negative. And then when the potassium channels close, we reach resting again until another action potential comes. So that's basically it for the contractile cells. Okay, so we have this long plateau necessary to keep our heart pumping efficiently and not to have too many heartbeats at one time. And it is the calcium channels that open slowly and close slowly that maintain this plateau. All right, so let's take a look at our next topic, which is our excitation-contraction. Here is that picture. So this looks really familiar, hopefully, because it's very, very similar to skeletal muscle. There's a couple differences. So when the action potential reaches your muscle cells, then, so here is our depolarizing current, right? Traveling very quickly through our gap junctions. The action potentials are going to create, first, a calcium channels opening in the plasma membrane. So we have calcium channels in the plasma membrane, and calcium is going to come into the cell. Now, that calcium is actually going to induce more calcium being released from the sarcoplasmic reticulum. So you can see that's happening here. So the calcium comes in from the outside, the extracellular fluid. It also comes in from the sarcoplasmic reticulum. So we have a high concentration of calcium, and this is where it's exactly the same with skeletal muscle, where the calcium binds to troponin, moves the tropomyosin out of the way, myosin heads can bind, and we have power strokes to create that, to create the contraction of the heart muscle. So the striations of cardiac muscle is due to the same organization of sarcomeres in skeletal muscle. So we have all the steps that are pretty much the same here. And at the same time, we have our active calcium pumping the calcium back into the sarcoplasmic reticulum. And also we have here the calcium getting pumped out of the cell, so we can maintain that concentration gradient in both areas. When calcium is pumped into the sarcoplasmic reticulum or out of the cell, the calcium levels drop, and then we have muscle relaxation, right? Because it's the same mechanism as skeletal muscle. So that's all we need to talk about. So not a whole lot of new information there, hopefully. And now moving on to the ECG. So I think that's it, yeah. So the ECG is a way to measure the heart's electrical activity. So what's coming from, so we're measuring that depolarization of the SA node, the AV node, and the fibers and the Purkinje fibers. And the way that they put these leads on your body is to measure different aspects of the heartbeat. So you can see that there's actually 3 leads here. There's lead 1 that goes from arm to arm. There's lead 2 that goes from your right arm to your left leg. And there's lead 3 that goes from your left leg to the left arm. Now, we're only going to look at what's happening in lead 2. This is the most common one to give you that sort of regular heartbeat wave, even though all these leads will give you a variation of the same kind of wave. So this is our ECG, and you're going to want to know basically what each little bump means on the ECG. So first of all, we'll start with the P wave. So the P wave is a small depolarization wave, and that's going to be depolarization of the atria in response to the SA node, right? So the SA node is going to fire, and then all the atria are depolarizing. Okay, so realize that depolarizing is not the same as contracting. So look at that in just a second. Then we have a little interval while the AV node is allowed to, it's delayed a little bit, and the ventricles are filling. And then we have a huge QRS complex. So the QRS complex is the depolarization of our ventricles, right? Because our ventricles are the most massive part of our heart. They're going to give the biggest wave of depolarization. And then our last little bump here is the T wave, which is when our ventricles are going to repolarize. So there is a bump for repolarizing. Why don't we see atrial repolarizing? So we see atria depolarizing, we see ventricles depolarizing, we see ventricles repolarizing, but we don't see the atria repolarizing. And this is because it's masked, it's happening at the same time as this massive QRS wave, so it's masked. We don't see it. So the atria are actually repolarizing here. Okay. All right, so we have another segment here. The ST segment is the beginning of the ventricle repolarization, and it should be flat. So let's take a look at the picture. And I have a note here. It says, note that the flat lines are during muscle contraction. There's no the electrical activity has already been sent to that muscle cells, and the muscle cells are responding by contracting. And remember, this is going very, very quickly. So let's start here, right? We'll start at number 1, where we see a flat line. Okay, so there's no electrical activity. A flat line is also known as the isoelectric line. There's no electrical activity, so it's kind of just at a pause. And then our SA node is going to fire, and our SA node is going to show up as our P wave. Okay, so you can see that SA node is depolarizing. The second, so take a look at what happens at number 3. The atria are going to be contracting, and look at where our purple color is. It's very small, but we have a flat line because the electrical activity has already been sent, but the atria are reacting to it by contracting. Next, we have our massive depolarization, right? So our AV, our bundle of His, our Purkinje fibers, that's all electrical activity, a lot of electrical activity showing up as our QRS wave. Then our ventricles will contract. But since the electrical activity has been sent and it's gone, it's a little flat line. So the ventricles are actually in contraction after they received that electrical signal, okay? So then we have another electrical signal, this bump, when the ventricles are repolarizing. And the reason it shows up is because it is some electrical, it's a switch in membrane potential for all the cells there. And then there we have it. And then we're back at number 1, where we have this pause of electrical activity right before the SA node fires again. So hopefully that you see how all that makes sense, those little flat areas along with those bumps. And the bumps really mean electrical activity, right? It doesn't mean that anything's contracting. It just means that the action potential is there. So the usefulness of ECGs is to detect normal versus abnormal heartbeats and to kind of figure out what's going on with the heart. Before we get there, let's just talk about a couple segments. So the PQ segment, let's go back to here, right? The PQ segment, which is actually not on this one, but the PQ segment is going to be the AV nodal delay. So this is that delay before the AV node fires. And then the RR interval, so I don't have a picture of that, but you see the R, so this peak is known as the R. So from peak to peak, that is going to be 1 heartbeat. And yeah, that's all I need to cover. Oh yeah, so this is a nice, well, this is the abnormal ECGs, but you can see peak to peak would be 1 heartbeat. So these are just examples of what could happen. I don't expect you to know or how to read ECGs. You have to be a specialist in order to know how to read ECGs really well. I want you to know what those waves stand for, what a normal ECG looks like, and then just some ideas about what could go wrong and some I just listed a few different abnormalities. So again, letter A, so we're number 5 under ECG arrhythmias. Arrhythmia means an abnormal heartbeat. Keep in mind that letter A is sinus rhythm is just your normal heartbeat. So this is normal. This is going to be your sinus rhythm. Okay. And tachycardia is a faster than normal heartbeat. So sinus tachycardia is seen here. Sinus bradycardia, a slower than normal heartbeat. So tachycardia is normal, right? If you're excited, if something's very stressful, your heart rate can raise. And then bradycardia, sort of less than 50 beats per minute. It can be a sign of problems, but it also can be a sign of conditioning. So if you're in really good shape physically, your heart doesn't need to beat as fast to push the amount of blood through your body for what it needs. So bradycardia can actually be a sign of conditioning if you're in good shape. And then I have just a little snippet that Lance Armstrong had a resting heart rate of 35, which is very, very low. And then there's things called heart blocks. So heart blocks is where the conduction system is blocked. So usually from SA to AV or AV down the line, there's a blockage in that conduction system. So first degree heart block is where you just have a slowing of the conduction through the AV node. And it's slow anyway, but this is just an even slower AV node conduction. And it increases the PQ segment because that PQ segment, again, was that segment of the AV node delay. And so it's going to sort of delay the atria contracting. You'll have a longer pause before the ventricles contract. In a third degree heart block, which is what we see here, again, don't expect you to recognize these ECGs. You have a complete loss of SA-AV node connection. So you have a loss of conduction through the AV node. The P wave becomes detached, right? Because the P wave is the SA node firing, and that becomes completely independent of the QRS. So remember that if the SA node does not reach the bundle can actually produce its own heartbeat. It does contain autorhythmic cells. It's just slower than normal. So if you completely detach the SA node, your ventricles will still contract in an independent and very slow manner, which might be too slow to sustain life. So this is actually a really big problem. And then we have fibrillation. So fibrillation looks really, really scary. You can actually have two kinds of fibrillation. Fibrillation just means an uncoordinated heartbeat. And you can have atrial fibrillation, which is not as bad and not as deadly as ventricular fibrillation, because if your ventricles don't contract, you are not going to get blood pumped throughout your body, and that is going to be your heart attack. So heart attacks are usually caused by these ventricular fibrillations. And just a note on defibrillators, right? So this is why we have a device called a defibrillator, because fibrillation is the term used for an uncoordinated heartbeat. So if you have, again, atrial fibrillation exists, it's serious, but it's not going to be an immediate danger to your life because the atria, while it's uncoordinated, are not responsible for pumping blood to your body. Ventricular fibrillation is a medical emergency, and if it happens in the hospital, this is what a code blue is. And you basically cannot have your heart in this state because you're going to not pump blood to your organs, the most important organ being your brain. So if your brain is going to have a few minutes of oxygen deprivation, that can lead to death. So you use a defibrillator, and a defibrillator basically stops the heart. It jolts the heart, pretty much resetting it, right? Stops the heart so that the SA node can trigger a normal conduction cycle. And sort of that's the hope, is to sort of zap the heart back into a regular heartbeat. All right, and because they are very successful, they are put in areas and sometimes in public places where you can access them. All right, so let's look at our cardiac cycle. All right, so the cardiac cycle. We're going to look at the phases, and this is just what's happening to the heart in sort of one heartbeat. And so we can take a look at this. I actually, I think I messed up the numbers anyways. So let's take a look at phase 1. So this is what your book has. Okay, so let's take a look at this first. So phase 1, which is here, phase 1, this is where the ventricles are filling. So if you look at your outline, the ventricles are filling, but it's both the atria and the ventricles are filling, right? So phase 1 is everything is in diastole. So remember that the word diastole means relaxation, and the word systole is contraction. So you can have atrial diastole, atrial systole, and ventricular diastole, ventricular systole. So the very first phase is going to be both of these chambers are filling. So both of them are relaxed. They're both in diastole. Blood is flowing into the right atrium from the superior and inferior vena cava, as well as the coronary sinus. This is a anatomy review. Blood flows into the left atrium from those pulmonary veins. And during this time, the AV valves are open. So you can see the blood is coming in, the AV valves are open, but the semilunar valves are closed, right? So the semilunar valves are not going to be open because we want the blood to fill those chambers. So phase 2, all right, so we have filling. The second, and then at the end of phase 1, we have a slight atrial contraction. So actually this is listed on number 3 under phase 1. At the start of the atrial systole, the ventricles are normally filled with about 70 to 80% of the blood. So this filling process already gives you 70 to 80% full ventricles. And then at the very end of when the ventricles are relaxed, you're going to get that final push, that little kick from the atria when they contract, just to push that extra 20 to 30% into the ventricles. In terms of pressure, right, the atria pressure rises slowly as it fills with blood. The ventricles they have low pressure because they're not contracting. They're just kind of relaxed and they're filling with blood. So the pressure inside is not very high. And then there's a small rise in the ventricles at the end because the atria are contracting. They're pushing the rest of that blood into the ventricles. Okay. So phase 2 is going to be systole. Okay. So in this phase, there's 2 phases of systole. So 2 and 3 are both where the ventricles are contracting. But the first step, number 2, is very short. And that's basically where your ventricles are beginning to contract, but they're not contracted enough to actually push the blood past those semilunar valves. They're just contracting enough just to get, just to close the AV valves. So you can see that the AV valves are now closed in 2 and 3. And also in 4, they're closed because the pressure inside the atria is growing, and that increase in the pressure of the increase in the pressure of the ventricles, right? As the ventricles contract, the increase in pressure is going to push up on those AV valves, and that's going to close those AV valves. So that first initial part where there's actually no blood ejecting is called isovolumetric contraction. So isovolumetric, the volume is the same. There's no change in volume. The blood inside the ventricle stays inside the ventricles. It's not going anywhere. And then number 3, which is the longer systole phase, is going to be where the ventricles are ejecting the blood. So there's a lot of pressure, so much pressure, it overcomes the semilunar valves. The semilunar valves are opening. And then it's going to push the blood through the aorta. It's also going to push the blood into the pulmonary trunk. So I have in letter C under phase 3 that the ventricular pressure is oops, it should be the other way. The ventricular pressure is greater than the aortic pressure in the pulmonary trunk. All right, so then we have the ventricular pressure falls, and then the atrial pressure falls until later, until the late systole. So the atria, they don't have any blood inside, so they're going to just basically be relaxed, and so there's not a lot of pressure there. In phase 4, the isovolumetric ventricular relaxation. Isovolumetric, again, just means that there's no blood traveling anywhere, that the ventricles are relaxed. They're in diastole, and both of our valves are closed. So you can see here, we have another situation where both of our valves are closed. So no blood is going anywhere. So that same concept about isovolumetric, no blood's going anywhere. And the reason why the semilunar valves are closed at this point is because there's pressure coming back from the pulmonary trunk and also the aorta, and so that pressure coming back in is closing those valves. That pressure coming in from the aorta and pulmonary trunk, that's called afterload. So looking at this, to wrap it up, we have our beginning of atrial systole. Actually, let's start at the beginning. So this is isovolumetric relaxation. Nothing's moving anywhere. Our valves are closed. Then we have our filling, right? So we have both atrial diastole and ventricular diastole filling with blood. The last part of that, the atria are going to contract just to get that last kick of blood into the ventricles. Then the ventricles are going to contract. The first phase is very quick. It's a very fast isovolumetric contraction. There's no blood going anywhere, just enough pressure to close those AV valves and not enough pressure to open the semilunar valves. Then once that contraction increases, we're going to have ejection. So the ejection of blood away from the ventricles, and the power of that ejection opens up the semilunar valves, and it remains, those AV valves remain closed. And then when it's done, it's relaxing, then that pressure coming back, so the pulmonary trunk, you see the arrow comes down this way, and then the aorta, the same thing, the pressure comes back. So that backwards pressure, when the ventricles have been relaxed, is going to close our AV valves. And then these guys are also remaining closed. No blood's going anywhere. And then again, when we hit that road again with opening, and both of our chambers are relaxed to fill. So that is the phases of the cardiac cycle. All right. If you look at this picture, so this is just this image here blown up, right? So this is just another way of looking at that same concept. Just looking at our phases here and what is open and closed. It should make sense to you based on just your knowledge of the cycle, what valves are open and closed based on the pressure. But I'll go over it one more time. So phase 1, we have the chambers, both chambers are relaxed, and our AV valves are open because both our atria are filling with blood and our ventricles are filling with blood passively from the atria. So the AV valves are open. And the aortic and semilunar valves are closed. Why are they closed? Is because that backwards pressure from the aorta and the pulmonary trunk there, because that blood slides back. If the heart's not contracting, the pressure of the blood slides back onto the heart. So the semilunar valves are closed. Number 2, this is where we begin systole. Okay, so 2 and 3, ventricular systole. We're not forceful enough to actually push through those AV valves. I'm sorry, the semilunar valves. So the semilunar valves are still closed, but it's enough to close those AV valves. So the AV valves are also closed. Then when the ventricles contract even harder, true ventricular systole, where you have ejection, right? So 3 is where we actually eject the blood. The AV valves are still closed because the force of that pressure pushes up on those AV valves and the blood, and that closes those AV valves, and it's enough to open up our semilunar valves, and blood is actively being ejected. And then we have relaxation or isometric relaxation. number 4, right? And again, both of our valves are closed. No blood's going anywhere, closed and closed, as our ventricles are relaxing. So hopefully that makes sense to you. We can see that we have peak atrial pressure in the middle of our ejection phase. All right, so the next topic here is just the aorta, right? The pressure in the aorta, because the aorta is our first blood vessel that is going to feed the system, right? It's going to be the first one that receives the blood for the systemic circuit. So in the aorta, in diastole, when the ventricles are filling, the aortic valves are closed because the blood pressure, again, from the aorta, it's moving back towards the heart. So the aortic valves are closed. And blood is still leaving the aorta. So this is right after we have that big push. So what happens is the blood, it's still leaving the aorta, and the pressure falls. And the lowest point of your aortic pressure is called the aortic diastole. In systole, the aortic valves are open, and the pressure rises rapidly with ejection. So you can imagine that you're pushing all this blood through this aorta. The pressure on the walls is very, very high. And the highest point of this is called systole. Okay, so if we look at the blue line, right, the aortic pressure, when the heart is relaxed, the aortic valve is closed, and there's this backwards pressure on the semilunar valves, so the aortic valve is closed. The aortic pressure starts to fall, right? So the lowest point is the diastole. And then when the heart is in systole, number 3, right, we have the ventricles actively contracting. The aortic valve, of course, it's open. And then we have the greatest pressure. So this is our systolic pressure, right? So we have diastolic pressure and systolic pressure. So the diastolic pressure is the highest pressure on the walls. So the aorta is very elastic. If you remember, it's an elastic artery. So it has a lot of elastic fibers, and so it can really expand with that pressure through ejection, and then it recoils. So right after that ejection, it's going to recoil back, and that aorta is going to push on the blood that's inside of it to even give it more oomph on the way out, more push on the way out through the aorta. And that also will splash back on the aortic valve. And when the aortic valve closes, it causes a little bump in pressure. Because imagine a wave coming back towards like a wall, right? It's going to hit that wall and then kind of create, if it's a tube, it's going to hit that wall and then kind of splash to the sides. So you have a little uptick in pressure right when the valve's closed. Anyways, that's called the dicrotic notch. That's why you have a little peak there. Now, the next topic is just the mean arterial pressure. So this is the average pressure that's in the in the aorta and arteries, but it's the average pressure that's there, and that's going to be the driving force that pushes the blood through the systemic circuit. All right. Okay, so number 4, I just mentioned that the walls are elastic to allow a constant blood pressure. So we can see that the blood pressure is actually pretty high compared to the pressure of other blood vessels. All right. Okay, so let's talk about our letter C, the volume inside of our ventricles. So what changes do we see in the ventricular volume? So we have some terms here that's going to come back. There is the end diastolic volume. So the end diastolic volume, or EDV, is the volume of blood inside your ventricle at the end of diastole. So that's going to be the fullest, right? At the very end of filling, your ventricles are at their fullest. So it's maximum amount of blood that fills. The end systolic volume is going to be where it's right after it's ejected all that blood, how much blood is left inside of the ventricle, because it doesn't empty completely. So the end systolic volume is what's left after systole, and that should make sense. Now, the difference between the two is how much blood left, right? You have a full ventricle, you have ejection, you have a little blood left in that ventricle. Well, how much blood left that ventricle? And that's going to be your stroke volume. So stroke volume is your fullest point minus what's left over. And so that's how much left. So that's stroke volume. So hopefully that makes sense to you. Now, there's another term called ejection fraction. And ejection fraction is the fraction of end diastolic volume ejected. So the fraction of your end diastolic, the fraction of how much you can actually fill your heart with during a heartbeat. So if you look at the notes here, the ejection fraction is the stroke volume over the end diastolic volume. So how much left versus how much was possibly inside of your ventricle. If you do a division, divide the two, you get a percent. And the percent in the notes is 54%. So let's say we have 70 mils is how much blood left your heart. It's the stroke volume. Divided by 130 mils, which was how much your ventricle actually had. And then if we divide the two, you get 0.54, which is 54%. So your heart, the ejection fraction is it could pump out about 54% of what your ventricle can handle, right? And that's normal. So it turns out that normal ejection fraction is 50 to 70%. And I have some other numbers for you, right? If you have low ejection fraction, 41 to 49, mildly low, you may not have any symptoms, but guess what? You might have problems during exercise. Your cells, because heart delivers oxygen, so your cells might get low on oxygen. Moderately low, very low. If you have a very low ejection fraction, that means your heart's not able to pump out as much blood as your body needs, and that can be very dangerous. All right, so that's ejection fraction. Heart sounds, last one before we wrap this part up. When you listen to someone's heart, you hear lub, dup. So those are sounds of the valves closing. And I'm not going to talk about specifics here, just know that the first heart sound is a lub. It's usually the softer of your heart sounds, and those are the AV valves, right? In the whole cycle, right? And then our second heart sound is going to be the dup, and this is going to be your harder sound here, and this is going to be the semilunars closing. Okay, so that's it. And it correlates with phases 2 and 4. All right, some review questions before we end part 1. When the ventricles are contracted, what is the state of the valves? So go ahead and pause it right now. So the ventricles are contracted. So this is in full systole. Okay, so we should see that the bicuspid and tricuspid valves are going to be closed, but the semilunar valves are going to be open. So that's going to be letter D. Which two types of autorhythmic cells coordinate and provide rhythm to the heartbeat? All right, so if you looked at your choices, it's going to be letter B. All right, next one. The influx of which ions account for the plateau phase of the cardiac contractile cells? Plateau is responsible because calcium is coming in. Which portion of the ECG corresponds to the repolarization of the atria? Look at your choices. The answer is letter D. What happens during the QRS complex? The answer is letter C, ventricular depolarization and atrial repolarization. What is the mean arterial pressure? The answer is letter A. What happens during the isovolumetric contraction? And that answer is letter D.